everyone, I am Dr. Garma Gupta, Assistant Professor at Sri Ram Swaroop University, Lucknow. So, together we will be exploring the knowledge or the field of developmental biology. So, the first question arises into our mind that what is developmental biology? Basically, development means that from the starting phase up to the end phase, whatever things occur in the whole process is developmental biology. For an example, if we see a plant develops from a seed, then from seed a root sprouts and a stem came out. Then from stem leaves come out, then leaves start preparing food and it forms various other things like bud, then flower, and then from flower fruit develops, and again this fruit contains the seed, and after giving the fruit, after some time the plant may die or it has a particular time period after which it goes under senescence or death. So this whole cycle from birth to death is called as developmental biology. Or not only birth, even before birth, that also includes under developmental biology. So the developmental biology can be divided into two parts. First is prenatal development, that is the embryonic development, and the next one is post embryonic development, that is the postnatal development. Embryonic development means that before the birth, whatever things are happening or whatever the process happening, that comes under prenatal development or embryonic development. And post embryonic or postnatal development means that after birth and up to death, whatever process is occurring, that will come under this. So this whole process, these two processes combines and forms the development of an individual. So all the things which we studied regarding this will come under developmental biology. Now, what is developmental biology? This is the study of process of development of adult organism, which can be basically the ontogenic development. Ontogenic development simply means the growth, differentiation and development of an individual which is multicellular in nature, and this development may depend on two things. First is genetics, that whatever genes it is carrying, and the other thing is environmental factors, because environmental factors play an important role on the development of an individual. So if two organisms have same genetic material but their environment is varying, so you can find out the changes between the two. Now the development occurs through the process of reproduction, and the ultimate aim of reproduction is to ensure the survival of genetic lineage or the ancestry or the genes which are carried out by the ancestors, and that has to be passed to the next generation. The development process can be divided into different types. First is embryogenic development. In this, sexual reproduction occurs by the fusion of male and female nuclei, and this leads to the formation of zygote. In this, the chromosomes of two parents get segregated, and then the process of recombination occurs. So the process of recombination makes different individuals or different offsprings which are non-identical or different from each other. So no offspring is identical to each other or to their parents, because they are taking the chromosomes from two different parents, and this is forming the different recombinations. Although in case of identical twins, the DNA or the genetic material of both the individuals or the identical twins are totally same, but they may vary because of the environmental factors involved during the growth process. So environmental factors also play an important role in making the difference between the identical twins or the monozygotic twins. In case of zygotic or fraternal twins, obviously the genetic material would be different, so they are totally different. But in case of identical twins, the genetic material is same, but the environmental factors are leading to different changes, so they may have different kind of features or some other characteristics. So all the offsprings are totally different from each other. Next is blastogenic development. In this, asexual reproduction occurs through somatic cells of parent body, like for example amoeba. The next is parthenogenesis. Parthenogenesis involves the development of a female and rarely a male, which develops through the gamete cell without fertilization. This occurs in case of lower plants and invertebrate animals like rotifers, aphids, etc. And it rarely occurs in case of higher vertebrates. In this, egg may be either haploid or diploid, or the organism may be obligate or facultative for parthenogenesis. If it is obligate, then there won't be any kind of sexual reproduction, but if it is facultative, so there might be chances of sexual reproduction, as well as at some stage it may be parthenogenetic also. So these are the different types of development. There are some other development also, like regeneration of missing body parts, metamorphosis of larval animals into adult, then continuous differentiation of new functional cells from stem cells. Now embryology, which is an important part of developmental biology. It is the study of development of cell stage, that is zygote, to the end of the embryonic stage till birth. Now what are the different divisions of embryology? First is descriptive embryology, which describes the developmental sequence of any embryo. Then comparative embryology, which is comparing the ontogenic development of different vertebrate organisms. Next is experimental embryology. In this, various developmental hypotheses are tested using genetic and embryonic manipulations. Then chemical embryology, it includes all the studies which involves various biochemical, biophysical, physiological techniques for understanding the sequence of embryological phenomena at molecular level. Now the next one, that is teratology, which is very important. It is the study of malformation or birth defects in an individual. There are various substances which are involved in causing birth defects, and they are called as teratogens. So in this, teratogens are also studied. There are certain examples like phocomelia, in which the arms of a child are poorly developed. The next is ectomelia, in which the arms are less developed in the child. Now the other one is evolutionary biology, embryology. In this, it is studied that how the development may cause changes in the evolutionary process, and how an organism's ancestry may not allow the changes that may be involved in the evolution process. So there are certain genes which do not allow the changes to occur, and it remains throughout the generations. So in this, basically the development biology is studied from the point of view of evolution. Now the next one is experimental embryology. In this, basically various fundamental mechanisms involving different processes like fertilization, cleavage, gastrulation, embryonic induction, determination and differentiation, these are studied, which are very important part of developmental biology. Now the next one is behavioral embryology. In this, the early development of nervous system and behavior is studied of a particular individual, and then what effect it may cause on the later stages of neurobehavioral development. So the effect of initial development of nervous system is studied to understand the later stages development of neurosystem. Now the next one is analytical embryology. In this, modern equipments are used to study the process of embryology, after which molecular embryology comes. In this, differential expression of inherited genes are studied in different population of cells. And the last one is ecological development. It studies the various environmental hazards or various environmental factors which are affecting the development of an organism and how it may make changes in the environmental regulation. There are various environmental regulations which are there for the protection of environment. Next one is importance of developmental biology or the applications of developmental biology. There are various applications of developmental biology in several fields, and which we will see now. The first one is in vitro fertilization, that is IF. There are lots of couples which cannot produce the baby. They are infertile couples. So this artificial insemination process can be used for producing a baby. Apart from that, in case of animals, the same process of IVF can be used to get the best animals. Either it could be in the form of best milk production, best meat production, or for any characteristic. Next one is teratology, which we have already discussed. So it features the possible causes of birth defects, which could be either some environmental agents like some chemical, viral infection, or some radiation or anything. So this is studied under teratology. Now in case of birth defects, there are some chromosomal birth defects also, like Down syndrome, which is due to the presence of an extra chromosome, and this is very common abnormality, and it could be also due to the mutation of genes that control the development, and it can be detected by amniotic fluid. This process is called as amniocentesis, which is used for the identification of any malformation in the DNA of parents. After that, it could be very important for understanding the human genome sequencing, human genetic manipulation, or cloning. It may also be helpful in exploring the various serious ethical and legal problems associated with human genome cloning and sequencing. Now the next one is proteomics and genomics. In this, various genes and gene products can be identified which are involved in the development process, and this may also be helpful in identifying the various diseases and its functions. Now the very important field is pharmaceutical industry. So developmental biology may be helpful in designing drugs for cancer, against various degenerative diseases like diabetes, arthritis, neurodegeneration, etc. And the other one is therapeutic targets, which is very highlighted nowadays. Basic understanding of various genes and signaling process involved in developmental biology or development sequence can be used to identify various therapeutic targets. It is also useful for combinatorial chemistry to create various drugs. Now the next one is model organisms. There are various developmental processes which involve various signal transduction pathways. So these signal transduction pathways can be studied easily in various model organisms like xenopus, zebrafish, Drosophila, or C. elegans. Now the next one is genetically modified animals. So these GMOs can be used for understanding the pathological mechanism of a particular pathogen, that how a particular pathogen is causing disease in the individual. It can also be used for testing the new experimental therapies like use of hematopoietic growth factors, erythropoietin, and GM-CSF, that is granulocyte macrophage colony stimulating. There is also application in the field of tissue engineering, stem cell therapy, and agriculture. In agriculture, basically it can be used for the production of some vaccine or some drug in the milk or eggs. So this was all about the introduction to developmental biology. Now the next lecture would be on history of developmental biology. Thank you so much for your attention.